Hey everyone, welcome back. All right, so this video is going to be about the anatomy of the spinal cord. All right, so first I want to just mention before we get into details where your nervous system develops from embryonically. So when you're a tiny little embryo, your tissues get divided into three different layers, and those are the ectoderm, the mesoderm, and the endoderm. The ectoderm is the outermost layer of tissue that gets developed. The ectoderm is what's going to turn into eventually your integument and also your nervous system. All right, so moving into the central nervous system here a little bit. So here's a diagram that shows your brain and your spinal cord, which is your central nervous system. And I just want to note here that your spinal cord, if we focus in on that portion, is very well protected. So the spinal cord itself is surrounded by lots of layers of protective tissue, including your vertebrae. All right, so when we talked about the skeletal system, we talked about how there are vertebral foramina in each one of your vertebrae. And the combination of all those vertebral foramina stacked on top of one another creates a canal basically for your spinal cord to rest in, where it's going to be completely surrounded and protected by bone. All right, so in addition to the bone of the vertebrae that are surrounding the spinal cord, we also have some layers of protective tissue called meninges. So the meninges are these protective layers of tissue that surround the spinal cord. They also surround the entire brain. So all of the central nervous system, brain and spinal cord, is surrounded by three meninges. All right, so here's a close-up view so we can see those meninges. So the outermost layer is called the dura mater. So the dura mater surrounds the entire spinal cord and brain. This is the outermost, most superficial of your three meninges. The next layer in is called the arachnoid mater. So arachnoid mater is the next one. And then the deepest layer, the one that's actually touching the brain and spinal cord, is called the pia mater. And the pia mater is very delicate, very thin layer that's actually touching the brain and spinal cord. All right, so again, dura mater is the outermost layer. Dura means tough, so this is the kind of thickest of those three layers, surrounding the entire thing, very protective. The middle one, again, the arachnoid mater. So this one is deep to the dura, but superficial to the pia mater. And between those two, between the arachnoid mater and the pia mater, is an area called the subarachnoid space, or the SAS. So subarachnoid space is where we find your cerebrospinal fluid. So all the cerebrospinal fluid that's kind of like bathing your entire central nervous system is found in that subarachnoid space. And then the deepest layer, the pia mater, a very thin translucent layer that's actually adhering to the surface of the brain and spinal cord. Okay, so between and around all of these different layers, we've got spaces. So I mentioned the subarachnoid space, but in addition to that, the space above the dura mater, so between the actual vertebrae and the dura mater itself, is called the epidural space. So the epidural space is the space between the actual bones of your vertebrae and the dura mater. And then you've got the space between the dura mater and the arachnoid mater, and that's called the subdural space. So epidural space above it, subdural space below the dura mater. Okay. And just a real quick mention, any sort of inflammation of the meninges is referred to as meningitis. So meningitis can have bacterial or viral causes, depending on what microbe is causing the meningitis. You could have kind of mild symptoms, or you could have very severe and life-threatening symptoms that associated with meningitis. Okay. Quick look here at epidural anesthesia. So probably a number of you have had epidural anesthesia if you've ever had a child. So the epidural anesthesia is when we inject the anesthetic into the epidural space. So again, the epidural space is the space above the dura mater, but below that ligamentum flavum, which is the lining of the inside of your vertebral foramina. So it has to puncture between the spines, the spinous processes of the vertebrae, and get into that epidural space. Right. So that's going to be a little bit different from a lumbar puncture. So lumbar puncture is going a little bit deeper than an epidural would. So lumbar puncture is when we need to get either we're injecting something into the cerebrospinal fluid or we need to extract cerebrospinal fluid. So, and again, to get to that cerebrospinal fluid, you have to get to the subarachnoid space. So down even deep to the arachnoid space is where we need to get for a lumbar puncture. So again, this is if we need to extract cerebrospinal fluid, which we might if we need to detect for presence of pathogens, if there's an infection here, or if we need to get some drugs in there. So here's where a lumbar puncture happens. It's usually between the third and fourth lumbar vertebra, so between L3 and L4 is where you would inject for a lumbar puncture. You can give anesthetics this way too, but because we're much closer to the actual spinal cord, you would need a much, much lower dose of the anesthetic than you would for, say, an epidural. Okay, so let's look at the overall cord here. So your spinal cord begins at the base of the brain. So brainstem comes off the base of the brain. Spinal cord will then leave the skull through the foramen magnum and extend down through all of those vertebral foramina. The spinal cord itself extends all the way to around between your first and second lumbar vertebra. So between L1 and L2 is a structure we call the conus medullaris. And conus medullaris is the official kind of end of the spinal cord. So if I highlight it here, so we go all the way from the base of the brain all the way down to the conus medullaris, and the conus medullaris is where the spinal cord officially ends. Now you still have nerves branching off past that, but conus medullaris ends the spinal cord at about L1 or L2. All right. So past that, so extending kind of beyond the end of the conus medullaris is a structure called the filum terminale. Filum terminale is basically an extension of the meninges. So the pia mater that was surrounding the spinal cord is going to continue past the end of the spinal cord, and it's eventually going to connect with the arachnoid mater and the dura mater, and all of those meninges are going to come together to form this filum terminale. All right. Also past that, we have a structure called the cauda equina. So cauda equina just means horse's tail in Latin. So this is a whole array of spinal nerves that are branching off of the end of the spinal cord. Okay. If we look at the cord itself, we can also see these bundles of axons that sort of branch off of the spinal cord, right? So the spinal cord is not there just on its own, right? It's connected to a whole bunch of peripheral nerves. And those nerves usually branch off of the spinal cord in bunches called roots. So the roots are groups of axons that are connected to the spinal cord. So we usually have a posterior root that's containing sensory axons, and then anterior, which will have the motor. All right, so here's a kind of schematic so we can see that a little bit better. So remember our pathway is that we have receptors, then we have afferent neurons, we have integrating neurons, and then efferent neurons that are going to trigger some sort of effector cell. So if we follow that pathway here, let's look at this. So we've got our receptor. That receptor is going to trigger an afferent neuron that's going to go into one of these peripheral nerves here. So here's a spinal nerve. We're going to transmit that signal through a dorsal root into the spinal cord. So here you're looking at a cross section of the spinal cord. That's going to cross a synapse to our interneuron. And then our interneuron is going to relay that message to an efferent neuron or a motor neuron. And that's going to go out the anterior side of the cord. It's going to find its way to an effector cell. In this case, it's a muscle cell. So we're going to get some motor movement as a result of that. All right, so we're always coming in through the dorsal side. So the afferent always comes in through the dorsal side of the spinal cord, and the efferent signal always goes out the anterior side or the ventral side of the cord. Okay, so looking at our central nervous system here, so we can see a cross section of the spinal cord and a frontal section of the brain here. We can see differences between the white matter and the gray matter. I think we've mentioned this before, that the white matter is where we find myelinated axons. So this is aggregations of axons that are surrounded by a whole bunch of myelin. And remember, the myelin is that protective covering that's made of lipids. So because it's made of lipids, it kind of looks a little white. Now the gray matter does not have myelin, so it doesn't have that same white appearance to it. Gray matter is usually aggregations of cell bodies, and the cell bodies or the somas of neurons are not myelinated. So we find a whole bunch of cell bodies, dendrites, and maybe even some unmyelinated axons in the gray matter. Okay, so if we look at a cross section of the spinal cord here, so here's an actual cord. Um, we can see the white matter and the gray matter. So we can see white matter is kind of associated towards the outside, and then we've got this sort of butterfly looking structure on the inside here. So we've got these little wings of gray matter on the inside. Right, so the wings of gray matter are going to be referred to as horns. So we've got posterior horns and anterior horns of these gray matters. And then we've got columns of white matter. So the white matter is referred to as columns. So we've got posterior column, anterior column, and lateral columns of white matter. And the way that you can tell whether you're looking at the anterior or posterior side or ventral dorsal side of the cord is by looking for this anterior median fissure. So anterior median fissure, I'll highlight it here, is this deep fissure on the anterior side. So whenever you see that kind of deep fissure, you know you're looking at the anterior side of the cord, and that would make the opposite side the dorsal side. Okay. All right, so the white matter of the cord is going to be divided into tracts. So remember that tracts are bundles of axons that we find in the central nervous system. The tracts are going to be either ascending or descending. I mean, they're either going to be moving up towards the brain or moving down away from the brain, depending on where we're going here. So afferent pathways or ascending tracts go towards the brain, and efferent pathways or descending tracts go away from the brain. Okay, and then here's another look at how the spinal nerves branch off of the spinal cord. So remember that I said that the posterior roots or the dorsal roots are where we have afferent neurons coming in, so afferent pathways. And then the anterior roots or ventral roots, that's where we have motor neurons or efferent neurons branching off of the cord. So it always goes in the dorsal side here, to an interneuron in here, and then out the efferent side on the other, on the anterior surface of the spinal cord. Okay, transection of the spinal cord. So transection means cutting it. Cutting it is never good. So that means that we've separated some of these tracks. So tracks that were either coming towards the brain or leaving the brain were severed, and that means that we can't continue to carry those messages past the point of transection. So transection can happen, during accidents. A lot of times, automobile accidents or things like that can cause a transection of the spinal cord. Depending on where we transect the spinal cord, you'll have different results. So if you transect the spinal cord way at the base of the skull, so way up here towards the bottom of your head, then that's going to result in death by asphyxiation because you just won't be able to get the messages to your lungs and your heart to continue to breathe and to oxygenate that air. I'm sorry, oxygenate the blood. If we cut the spine, if we transect it in the cervical area, that's going to result in quadriplegia, so paralysis, full-on paralysis. If we transect the cord a little bit further down, then you can get paraplegia or paralysis of just the lower half of the body. So it just depends on where the transection happens. Okay, now let's talk about how we get blood supply to the brain really quickly here. So blood supply to the brain comes through a few different pathways. So it's going to mostly come from your carotids. So carotids are the arteries on either side of your neck here that's going to be carrying blood up to your brain, and also through your vertebral arteries. So vertebral arteries go through the vertebra and up and supplying the back of the brain here. So all of those arteries are going to kind of branch together at the base of the brain, and we're going to get this structure that we refer to as the circle of Willis. So let me highlight that here. So circle of Willis is this sort of structure here. And that's where all this blood that's coming in to feed the brain kind of winds up in this circular area. And that's so that we can get collateral flow, that we can get lots of sources of blood to the brain, so that should something happen where one of these arteries might become blocked or narrowed, we've got other arteries that are also supplying the brain, because again, the brain is the most important thing. It's in control of the entire body. We need to be supplying it with a constant, fresh supply of oxygenated blood. Okay. And then cerebrospinal fluid. So remember that even though we've got all this blood coming to the brain, the blood, we also have that blood-brain barrier, right? So we've got those astrocytes that are surrounding all of the little capillaries in your brain that are helping to protect the brain. So we basically need to create a fluid that's going to help get the brain the nutrients and the oxygen it needs, but not have all of the other kind of possible toxins that might show up in the blood. So we need to create the cerebrospinal fluid. Remember that CSF is created by ependymal cells. So ependymal cells are, a lot of them line these ventricles, which are kind of cavities on the inside of your brain. So they're going to be filtering the blood and creating the cerebrospinal fluid in those ventricles. And then it's going to travel around in that subarachnoid space to sort of bathe your entire brain and spinal cord. Here's a better look at some of those ventricles. So you can see these kind of cavities in the brain here. Let me highlight them. So lateral ventricles. These are areas in the brain that are lined with choroid plexuses. So choroid plexuses would be this kind of structure over here. So these choroid plexuses are lined with ependymal cells. As blood flows through the capillaries near these ependymal cells, the ependymal cells are able to filter the blood and create cerebrospinal fluid from that filtered blood. All right, so it's basically filtering any possible toxins out, and then allowing you to create cerebrospinal fluid that's going to continue to circulate around in those ventricles and also in the subarachnoid space. Okay, now once we create the cerebrospinal fluid and it's supplying all of the tissues of your central nervous system, we also need to reabsorb it back into the blood. So we have to continue to circulate this and generate fresh CSF and also discard the old CSF. And we discard it by recycling it back into the bloodstream so that it can be cleaned out through the normal process that we clean your blood. So to get the CSF back into the bloodstream, it enters through the arachnoid villi. So arachnoid villi, you can see, are located right up top here. So arachnoid villi at the top of the brain is where we're going to allow that cerebrospinal fluid to re-enter the bloodstream so that it can be filtered and cleaned. Okay, so here we can see a little bit better. So the zoomed in cross section of the arachnoid villus, we can see where cerebrospinal fluid is going to re-enter the bloodstream. Okay. As far as the production of the right amount of cerebrospinal fluid, right, we have to make sure that we're producing enough to bathe all the tissues of the brain, but not producing so much that it's not that we can't resorb it into the bloodstream, right? So if we have some sort of blockage, if we're not able to drain that cerebrospinal fluid back into the bloodstream as well as it should be, then we can get this condition known as hydrocephalus. So hydrocephalus basically just means water on the brain. It means we're accumulating too much cerebrospinal fluid and it's not draining back into the bloodstream as well as it should be. Okay, a few questions here. So arrange the meninges from superficial to deep. So figure out which of those is most superficial, which is in the middle, and which is the deepest. And then tell me where we give an epidural injection, above or below what layer. And then what is the SAS? And then what do we find in the SAS? All right, so there's your answers. So the most superficial meninx is the dura mater, then we have the arachnoid mater, and then the deepest is the pia mater. Where do we give an epidural? So it's going to be above the dura mater, superficial to the dura mater. And then the SAS stands for subarachnoid space, and that space contains the cerebrospinal fluid. Okay, a few more questions here. Where does the spinal cord end? So about what vertebrae do we find the end of the spinal cord? Between which vertebrae do we perform a spinal tap? So if we have to go in for a spinal tap, which vertebrae do we look for to enter? What extends in the vertebral canal past the end of the spinal cord? So once the spinal cord ends, what keeps going in that vertebral canal? And then what is produced in the choroid plexuses of the lateral ventricles? All right, so answers here. The spinal cord ends between lumbar vertebrae 1 and 2. So between L1 and L2 is where the actual spinal cord ends. If we need to do a spinal tap, we're going in between L3 and L4. And again, that's because it's a little bit safer to go in to the canal past where the actual spinal cord has ended. So that reduces any possible damage to the cord. And then what extends in the vertebral canal past the cord, that's the filum terminale. And then what's produced in those choroid plexuses is cerebrospinal fluid. All right, so that's your overview of the spinal cord. I hope this was helpful. Let me know if you have any questions.